Okay, now we're going to talk about the adaptive immunity in Chapter 4. Hopefully you've had some time to look at the innate immunity before we dive into this one. That one's a little bit more simplistic and easier to understand. With the adaptive immunity, we bring in different types of cells with different types of cellular communication that happens within those. So here we go. All right. The first thing we're going to talk about is an overall view of the adaptive immunity. So we talked about the innate immunity where the baby is born. Well, now mom brings the baby over to Auntie Ethel's house and she loves the baby and kisses the baby, and before you know it, the baby has some exposure to some bacteria. Okay. The adaptive immunity is able to remember that prior exposure. So the baby will be exposed and then start to create that memory response. This increases response to a pathogen upon repeated exposure. So if Auntie Ethel comes over next time, baby's body may remember that. It takes longer to become activated, but is longer lasting. So think about when you get a vaccination or if you get the chicken pox, okay? It's going to take a little bit longer to activate it, but that immunity lasts pretty much the rest of your life. So your body has that ability that after it's adapted to maintain that immunity. This one involves the T and B lymphocytes, where the innate involved more of the granulocytes. The first thing we're going to talk about is the double negative stage of T cell differentiation. This is where the chemokines drive the differentiation process. So the chemokines are the chemical messengers that cause this to happen. Your thymocytes in your thymus undergo a rearrangement of genes coding for T cell receptors. And you see TCR here. Sometimes we're going to start to refer to some of these things with their mnemonics rather than the whole word. There's also a CD3 T cell receptor complex. This is what recognizes it as a T cell. It doesn't really have, a role in life yet. The double positive stage is the next, where the thymocytes express CD4 and CD8 and rearrange gene coding for an alpha chain. Don't worry about that second part of the sentence, but this is where the double positive is the CD4, CD8 positive. That's what the double positive stands for, okay? Don't make it too complicated. This is positive selection if there's a functional T cell receptor. So here's where it can decide, do I keep this or do I throw them away? Okay. We have major histocompatibility complex restriction, where only cells that react with the host will survive. Negative selection is where cells that react with self-antigen then become destroyed. Okay. So if you have CD4, CD8, then you become more of a helpful type of cell in this process. So T cell differentiation, we'll talk about the mature T cells first. The survivors of positive and negative selection, means they made it all the way through the thymus in this process, exhibit either CD4 or CD8. Here's some easy ways to memorize this. CD4 T cells are T4 helper cells. CD4 is 4, help has 4 letters. So I think of CD4 as the help cell. Okay, it's got 4 letters. CD8 T cell are cytotoxic T cells that kill target cells. Cytotoxic has 8 letters. It really doesn't, okay? But we'll pretend it does. It looks more like it does than CD4, right? So if you remember the longer word, cytotoxic is CD8. T4 helper is 4, okay? So one helps and one destroys. We also have CD8s as suppressor cells as well that we'll talk about. So the cytotoxic and regulatory and suppressors all have a lot more letters. Those are the CD8 cells, okay? And what makes it really unfortunate, if somebody has HIV infection, it destroys the CD4 helper cells. We need the helper cells to help our immune system, right? We destroy those with the human immunodeficiency virus. Before you know it, all kinds of infections take over our body. So you don't die from HIV, you die from the other stuff that kills you. All right, we'll talk about these T4 helper cells first. They have that CD4 receptor, which we've talked about. They recognize antigen and class II MHC proteins. This accounts for 2/3 of the peripheral T cells. Okay, so you can see the CD4 or 8 on this T cell here binding. Okay, if it binds to self-antigen, there may be apoptosis. If it's a macrophage, dendritic, or thymic epithelial cell, it doesn't, because we don't want to destroy it. Okay, if it's our own cell, we usually don't destroy those. So then we have T helper subsets of this. We got the T helper 1 and the T helper 2s. Got to recognize the difference. The T helper 1s produce interferon, interleukin 2, and tumor necrosis factor B. They activate the cytotoxic lymphocytes and macrophages. On the other hand, the T helper 2 cells produce a lot of different interleukins, okay, not just 2, but all the rest of them essentially. It helps B cells produce antibodies. It regulates that B cell activity, okay? So there's a difference here. Be familiar. Might just require making note cards or something like that. So I just explained a lot of stuff, right? And in your head is probably just a big bunch of jumbled words. So I found this video, which I think is fantastic, where this guy does a really good job of explaining how these cells head into the thymus, come out of the thymus with CD4, CD8, and what they do after that. So it's T cell development made very simple. So this should help bring what I just mentioned kind of into fruition, so it makes a little bit more sense for you. And I apologize, it'll be just a second to get it running.
So, I know you're thinking to yourself, what am I going to do today? Let's learn about how T cells mature. T cell development. T cell development. Nothing more exciting. So we've got T cell development. What goes on? Well, we're initially going to have prothymocytes. So prothymocytes, so something before we get to a T cell that begins to develop in the thymus, and eventually we'll get different T cell lineages, so different T cells. So let's talk about how we get there. Initially we have this prothymocyte. This prothymocyte is a T cell precursor, and it's going to be made in the bone marrow. So it's going to be made in the bone marrow. It quickly develops and travels to the thymus. So it's going to be made in the bone marrow, but quickly develops and goes to the thymus. Because in the thymus, this is where most of the T cell development happens. B cells are going to mature in the bone marrow, while T cells will occur in the thymus. Easy, because B cells, bone marrow. T cells, thymus. Easy. T cells, thymus. B cells, bone marrow. But we're just going to talk about T cells right now, because this has an interesting kind of pathway. So once in the thymus, we've got these prothymocytes, and what's going to happen is we're going to have a cortex. So the cortex is going to be the outer layer, and the inner layer is going to be the medulla. So cortex is going to be outer, medulla is middle. And you're going to see that in the adrenal glands. You're going to see that in the kidney. The cortex is going to be the outside kind of capsule, while the medulla is going to be in the middle, M for middle. Okay, so what's going to happen? Our prothymocyte is going to travel to the thymus. It now becomes a T cell precursor. That T cell precursor is going to start in the cortex. So we're going to start out here, and it's going to work its way into the middle. So this thymus, it's going to be derived from the third brachial arch. So the third brachial arch is going to develop the thymus. The thymus is going to be bigger in children, and it more involutes as we get older. So the thymus will be very prominent in children. It's going to be more prominent in children. I'll leave it at that. Okay, so we've got a cell here. It's going to develop in the cortex first, and it'll work its way to the medulla. So we've got this T cell precursor, and it's got a T cell receptor, T cell receptor, TCR. And what it is it's going to be CD3 positive. It's going to be CD4 positive. It's going to be CD8 positive. Holy cow, it's got a lot of markers on it. It's got so much markers on its cell surface, it doesn't know what it's doing yet. So what do each of these mean? Well, let's start with CD3. CD3 is going to indicate that it's a T cell. That's all it means, is it's a cell marker for for T cells. CD4 positive means that's a marker for T helper cells. So all T cells will have CD3 on the outside. CD4 positive cells are going to indicate that the cell is a T helper cell. And CD8 positive means it's a cytotoxic T lymphocyte. So why does this one have all three? It's because it can become a T helper cell. It can become a cytotoxic T lymphocyte. So you want all the markers there because it can become these subtypes. So you'd expect that this, and the T cell receptors could be also found on all of the T cells. So we have two different routes that we can go. We can either become a CD8 positive or a CD4 positive. Also has the CD3 as well. I'm going to stop writing it in because it gets a little messy. It's also going to have the T cell receptors that I'm not going to write in as well. So what's going to happen is we're going to stop displaying the CD4 or the CD8 so we get just one instead of both. What this is going to do, we're going to get this through apoptosis or cell neglect. As the cell develops, as this prothymocyte develops, and now it's the T cell precursor, and as it develops, it's going to lose its receptors. If it doesn't lose its receptors, we're going to get apoptosis. Or we could get cellular neglect. So we're not going to develop it anymore. Eventually, the cell will die. Either way is the same. We're going to get rid of those cells. So the cells that express now just one are going to be good. So this is called positive selection. We are just making it so we have CD8 or CD4. We're getting rid of some of this other stuff that's attached there. So this is positive selection, and it's going to occur in the cortex. Kind of an important concept to understand. Positive selection occurs in the cortex. So we've got our T cell receptor, we've got our CD3 and our CD8. What's going to happen is it's going to go to the medulla. And once in the medulla, we're going to get something called negative selection. Negative selection is where we get rid of these T cells. So let's say we develop a whole bunch of T cells, but every 1 out of 1,000, one might be self-reactive. So we want to get rid of those self-reactive T cell precursors. Because if it's self-reactive, we don't want it to be circulating within the body, reacting with the body, damaging stuff. So we want to get rid of those. And that's where the negative selection comes in. And this is based on T cell receptor strength, binding strength. So in the thymus, if the T cell receptor binds really strongly to the antigens and the molecules within the thymus, you know that it would bind other stuff within the body really tightly. So if it's a really strongly binding T cell receptor on this cell and it binds the thymus really strongly, what's going to happen is we're going to need to get rid of it through the apoptosis or cellular neglect. We don't want cells within our body that react. Let's say the T cell binding is weak. That's good. We want a weak binding because that means it's probably not going to bind to the antigens present in our body. So negative selection rules out self-reactivity. Positive selection is getting rid of the excess cellular markers. We're differentiating our T cell precursors into CD8 positive or CD4 positive primarily. So once we've gone through positive selection, once we've gone through negative selection, we leave that medulla. So we worked in the cortex, then we went to the medulla, and now we're going to leave the medulla and go to a lymph node. And that's going to be the final location for our T cell. Now we have different types of T cells. We're going to have this cell that's, I'll draw every molecule out, TCR. We're going to have the T cell receptor, we're going to have a CD3, and this one will be a CD, let's say 8. And then we're also going to have another cell. This one will have a T cell receptor, still a CD3, meaning it's a T cell, and we've got a CD4 positive. All right, so we've got two different pathways. Now let's talk about what each of these mean. So the CD8 positive is going to be a cytotoxic T lymphocyte. This is going to mediate cell-mediated immunity. It's going to destroy stuff. It's going to find cells and kill them. All right, so it's going to bind the CD8 positive is going to bind MHC1. MHC1 is going to be expressed on almost all cells, minus red blood cells, but pretty much all cells in the body. And this MHC1 is going to take intracellular antigens or proteins or sugars or anything. It's going to chop them up, it's going to spit them out and present it on the MHC1 complex on the cell surface. And once on the MHC1 surface, our CTL is going to come along. It's going to use its CD8 positive binding to bind to MHC1. It's going to check is that molecule that's being presented on the MHC1, the stuff that was from the inside of that cell, is it good? And if it's good, then it's going to leave it alone. If it's bad, that cytotoxic T lymphocyte is going to kill that cell. So that's going to be the cytotoxic T lymphocytes. Now you've got the T helper cells. T helper cells are going to be the CD4 positive, while before it was CD8 positive for cytotoxic T lymphocytes. T helper cells, you may have heard CD4 positive always binds MHC2. Well, what does this mean? MHC2 is going to be found on antigen presenting cells, so like dendritic cells, for example. MHC2, so the dendritic cell is going to find antigen in the extracellular space. It's going to have arms, and it's going to grab antigen. It's going to internalize it through phagocytosis. It's going to chop up that antigen and present it on its MHC2. So it's going to present extracellular stuff on its MHC2. Likewise, we're going to have the CD4 binding site come along and bind to the MHC2 on those antigen presenting cells. We're going to check, is that stuff that it's presenting good? And if it's good, it'll leave it alone. And if it's bad, that's when our T helper cells come in. So you've got two different types. You've got T helper 1, we've got T helper 2. Two different types. T helper 1 cells, so T helper cells will differentiate into T helper 1 cells through interleukin 12. And then T helper cells will differentiate into T helper 2 cells via interleukin 4. So what are the two different types? Well, type 1 helper T cells are going to pump up macrophages. So they're going to release interferon gamma, and that interferon gamma is going to stimulate those macrophages to be super macrophages, and they're going to internally kill stuff. They're going to phagocytose stuff. They're going to work overtime shift. And then also they're going to pump up cytotoxic T lymphocytes as well. All right, so then we've got this T helper type 2. What this is going to do is it's going to help B cells. They're going to help B cells. And what they're going to do is B cells make antibodies. So the T helper T type 2 cells are going to help the B cells make antibodies. So you're going to have a stronger humoral response. So just in recap, what we're going to do is we're going to go from prothymocytes made in the bone marrow. We're going to have those B cell precursor or T cell precursors go to the thymus. They're going to start in the cortex. In the cortex, they're going to undergo positive selection. They're going to choose between CD4 or CD8 positive T lymphocytes. They're going to travel to the medulla. And in the medulla, they're going to undergo negative selection. And that's where they get rid of the self-reactive T lymphocytes within the body. We don't want self-reactive T lymphocytes in the body. And then from there, they go to the lymph node, and that's where they're active. And that's where we get the different types. So we're going to have the different types, and depending on what type it is, it's going to have different function. Hopefully you found this video useful. Tried to cover all the basics, the stuff that I struggled with when I was going through this material for the first time. Hope you found it useful. If you did, please click like. Always like some likes. I also enjoy reading your comments, so please leave lots of comments. Otherwise, Subscribe for more videos, and peace out.
All right. Well, hopefully you found that as helpful as I did. I thought he does a much better job of drawing because I am a scientific type of brain, definitely not an artistic brain. So when I can find somebody else to draw something out better than me, I'm certainly going to use it. So a couple other things that we'll talk about, the T regulatory T cells. These have CD4 and CD25. CD stands for cluster of designation. So it's just a name for some of these antigens that make these cells essentially what they are, okay? They suppress the immune system response to self-antigens, and they secrete inhibitory cytokines to inhibit proliferation of other T cell populations. Now imagine if the immune system just went totally crazy and there was nothing to slow it down. Okay, it'd be kind of like taking a big Chevy Suburban down Highway 41 with no brakes. It wouldn't be a good idea. You need to be able to stop, right? All right. Next, we're going to move on to the B cell differentiation. Now, our friend just talked a little bit about where that comes into play, but we're going to go into a little bit more detail, and I have some other great drawing videos. I know if you're anything like me, just reading all these words and listening to it, having that auditory type of learning and even reading is just overwhelming, but if you can see it, it really helps. So we're going to talk about it, and then again, you're going to see this happen with a drawing, okay? So the B cell differentiation occurs in the bone marrow. Remember he said T cell, thymus, B cell, bone marrow. Makes it pretty easy to memorize. It starts with a hematopoietic stem cell that develops into an early lymphocyte progenitor. We're going to start getting into some of these things in the hematology course that we're taking concurrently with this in most cases. And you're going to learn about those cells. They're just kind of like infantile cells that produce more lineages of cells, okay? It all starts with that. We have an early lymphocyte progenitor. It involves the development of the mature immunocompetent B cells. It activates the B cells with antigens, and it differentiates those B cells into antibody-producing plasma cells, which is, in the end, what creates all those wonderful antibodies we need when you are re-exposed to some type of an antigen or form, which would be a foreign organism. So here's a little picture of what is going on here. So we have that stem cell, that hematopoietic stem cell. And we're going to go through a whole series of cells before it becomes a mature antibody-producing cell. So it starts with pro-B cells in the bone marrow. Okay. It then turns into a pre-B cell. Then we go to an immature B cell before we become a mature B cell. The mature B cells are the ones that are in the spleen and lymph nodes before they enter circulation, which is then where they would make, the antibodies to help fight off infection. So the pro-B cells were the first ones. These developed from a rearrangement of genes that code for heavy and light chains of antibody molecules. Now you're going to yourself, what the heck does she mean, heavy and light chains of antibody molecules? Well, next week we're going to spend a lot more time looking at each of the parts and pieces of the antibodies, along with the chains that are put together to create them. So don't worry about that right now. We are going to get there. They must successfully rearrange one set of heavy chain genes to become a pre-B cell. So essentially this is just a stage to get us to the next one. Next, we have pre-B cells. These accumulate in cytoplasm. They express on cell surface with light chain. And again, you guys are kind of like, what the heck? Okay, so don't worry about that. We're going to get into these things later on. The next stage was the immature B cells. These have complete IgM antibody molecules on the cell surface. IgM is very large, and it can bind a lot of antigen. Okay. They have variable regions that determine the specificity. So the B cell with the antibodies are very specific to certain antigens, okay? So you don't have one type of antibody that binds all kinds of things. It's not like a natural killer cell that can go out and attach to anything. These are very specific for very specific antigen. And if mature B cells survive, they move on to the spleen. And they have other surface proteins such as CD21 and CD24, or CD40, sorry. Again, cluster of designation is what that stands for. Once we get a mature B cell, they develop in the spleen. Then we'll have the marginal and follicular B cells. The marginal B cells remain in the spleen. They're quickly to respond to bloodborne pathogens as needed. The follicular cells migrate to the lymph nodes and other secondary organs to just kind of wait around. Okay. They also exhibit IgM and IgD. IgM is very large. IgD is very small amount that we'll talk about next week. That surface immunoglobulin provides an activating signal to B cells when in contact with an antigen. So we have to have the antibody on the surface of these for this to happen. Once they do become stimulated, they enter an antigen-dependent phase, forming memory cells and antibody-secreting plasma cells. Okay. All right. So again, my art is horrendous, and I love handwritten tutorials. I don't know if it's his art, which is absolutely amazing. I don't have handwriting as good as him, but his voice too is great. Some of the ways he pronounces some things is a little bit different, which is kind of fun to listen to as well. All right, so let's watch this B cell maturation and how this process happens. This one's only 5 minutes.
Hi, in this tutorial I'm going to talk about the maturation of B cells. Now the first thing to recognize about B cells is that they go through various stages of development. So I'll begin by drawing up these stages. And during the process of maturation, a B cell will progress through all these stages. Now I'll label each stage. B cells arise from stem cells within the bone marrow. These progress to pro-B cells, and we'll look at the differences between the early pro-B cell and the late pro-B cell. These then progress to pre-B cells, and then onto immature B cells. The immature B cells then migrate to lymph nodes, the spleen, and other secondary lymphoid tissues. At this time, they then progress to naive B cells, and they eventually may become mature B cells. The first part within the bone marrow is the antigen-independent phase, because B cells progress through these stages without the presence of an antigen. The part within the secondary lymphoid tissues is called the antigen-dependent phase, because to progress from a naive B cell onward requires the presence of an antigen. So let's see how all this happens. This big cell up here is a bone marrow stromal cell, and it is required to send a series of signals to get the process started. Initially, the stem cell binds to the stromal cell via the binding of a protein called VCAM1 on the stromal cell to VLA4 on the stem cell. At this point, heavy chain DJ rearrangement occurs. If you don't know what I mean by this, see the previous video on antibody production and recombination. Once VCAM1 and VLA4 have bound, they promote the binding of a second protein coupling. This is the binding of stem cell factor, or SCF for short, on the stromal cell, and c-KIT. They cause the early pro-B cell to express the interleukin-7, or IL-7, receptor. At this point, heavy chain VDJ recombination occurs. IL-7 released by the stromal cell is now received by the IL-7 receptor on the pro-B cell. IL-7 drives further maturation of the pro-B cell. The genes for the heavy chain are transcribed and translated, and there is also downregulation of VLA4 and c-KIT, so the pro-B cell releases from the stromal cell. The translated heavy chains organize and present on the surface of the cell along with a protein complex called the surrogate light chain. This is known as the pre-B cell receptor, or the pre-BCR for short. At this point, light chain VJ rearrangement occurs. Then the light chain and heavy chain present together on the surface of the cell. This is an immunoglobulin or an antibody known as IgM. At this point, we call this cell an immature B cell, and this immunoglobulin can also be referred to as the B cell receptor or BCR. This B cell migrates to the secondary lymphoid tissue, where it expresses both IgM and IgD on its surface. This B cell will stay in this state until it meets its antigen, at which point we call the cell a mature B cell. And another whole series of events to fight a possible infection take place. And that's an overview of the maturation process of B cells. For more free tutorials and the PDF.
All right, so there was a little introduction of what happens as those B cells mature from the bone marrow out into the bloodstream and lymph nodes. All right, so where we ended with that were these plasma cells. There's a lot of they have a lot of cytoplasmic immunoglobulin, but little or no surface immunoglobulin when we get to this. One thing I want to point out, and we'll talk about this in hematology as well, is the look of a plasma cell. They're very dark blue. It is a lymphocyte. You can see a normal lymphocyte kind of over here to the left, but it has an eccentric nucleus. Okay, so it's kind of off to the side. And we've got this area of pallor next to the nucleus as well. So it kind of has a fried egg type of appearance. The big claim to fame is they produce antibodies. We find them in the peripheral lymphoid organs and the bone marrow. But once they've formed, they leave the lymph nodes and circulate in the peripheral tissue. They have a very short lifespan, and some migrate to the bone marrow for long-lasting immunity. So think of the little girl who got the chicken pox. Her body gained this immunity. Now she's going to have that long-lasting immunity to it. And a lot of those come from the plasma cells that are going to be in the bone marrow. So there is, going back to the role of the T cells and B cells in the adaptive immune response, we're going to just review a little bit here. The T cells are antigen-presenting cells that are activated during the innate immune response. Those T cells then interact with the antigen-presenting cells to initiate the adaptive immune response. The T cells circulate through the bloodstream, lymph nodes, and secondary lymphoid tissue looking for antigens. Okay. The action of these, okay, is where the exposure to the antigen presented by the macrophages causes the production of the CD25 receptors for that interleukin-2 that we mentioned earlier. That interleukin-2 causes the sensitized CD4 T cells to secrete cytokines, resulting in the T4 effector cells that have various functions. Some, and this is where it gets important, some CD4 cells secrete interleukins that recruit macrophages, activated T helper cells, whereas others activate CD8 T cells to increase cytotoxicity against viral infected cells, also enhance the antibody production of B cells. So they do end up working together in the end. The T cytotoxic cells, which are the CD8 T cells, recognize the foreign antigen along with the MHC class 1. When the binding occurs, the granules move towards the point of contact with the target cell. Those granules fuse with the membrane and release perforin. Perforin inserts itself into the target cell membrane and polymerizes to form a pore. Okay? The contents of the granules are released, triggering apoptosis of the target cell. Okay, so here's our perforin pore that we just mentioned. Now the role of the B cell in the adaptive immune response. Well, first the T-dependent antigens bind to the immunoglobulin receptors on the B cells. The antigen is processed and delivered to those CD4 T cells. The T helper cell binds by means of CD3 T cell receptor complex and delivers further activating signals through binding of the CD40 on the B cell to the CD40L receptor on the T cell, which you can see in the middle here. The cytokines are then released, you can see them up here, from the T cell, which enhance B cell transformation to plasma cells. Independent antigens can then bind B cells through immunoglobulin receptors and trigger B cell transformation directly. The antigens can also be bound to B cells' innate immune receptors, such as those toll-like receptors, seen here. We do have laboratory identification of lymphocytes, which can help diagnose malignancies, immunodeficiency diseases, and AIDS. So in malignancies, if we have abnormal lymphocytes, we're looking for certain cell markers in different malignancies, and we use something called flow cytometry, which we'll talk about here in a minute. I have a video. Immunodeficiency diseases, okay, so if you're missing some type of an immunoglobulin, or you have an abnormal amount of an immunoglobulin, these are all things we're going to learn about this semester. AIDS, we talked about. We start with HIV. HIV attacks the T4 helper cells, destroys them. Then we move to acquired immunodeficiency syndrome once those T cells are destroyed. We use flow cytometry to detect the different types of lymphocytes. It can segregate lymphocytes into different subsets, and we can attach monoclonal antibodies to certain types of lymphocytes and count them based on their exact type and subtype. So here's a little video that goes over exactly how a flow cytometer works. I can't exactly bring a flow cytometer into class and be like, okay guys, here's where the cells go through. They come through one at a time. We use a sheathing reagent to push the cells through. We add the immunoglobulin. This is just going to show it to you, okay? Again, my art skills are horrible, so me trying to draw this, you guys would be even more confused.
In this animation, we will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescence signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
All right, well, what I found really interesting about that was, and this didn't exist back when I was in school. I love that video. It can tell you exactly how many of each types of cell there are based on size and complexity. But also, if we're looking for a certain cell population, if we're trying to quantitate CD4 and CD8 T cells, we add a fluorescent antibody to that patient's sample that is specific to those CD markers. As it passes through that flow cytometer, it will count exactly how many of each type of cell there are based on those CD markers. So it can give us an exact count so we can tell the physician, especially if somebody's got HIV, how many CD4 cells they have versus CD8, right? Because you want the CD4s to be higher than the CD8s because you want more helping than suppressing or cytotoxic. So very, very, very useful. And it's actually very simple to look at when you look at it in terms of that video. All right, something I want to talk about here really briefly, and we'll talk about a little bit more when we get to the immunoglobulins, but I want to touch on it now because there is a little bit of it on your exam coming up in the next couple weeks. And I want to make sure that we get a good head start on this and that you know the difference. So this is what we're going to end with today. So the difference between active and passive immunity. Active immunity is actively giving somebody an infection, okay? It is getting a chicken pox or getting a shot for chicken pox. Okay, that's active immunity. You're actively getting that organism. Passive immunity is getting antibodies injected into you. Active is getting antigen injected into you. Okay, so that's the difference between those. Now let's look at the active immunity. Natural active is naturally getting the infection. It's back when I was 2, my mom taking me to my cousin's house because she had chicken pox to play with her so that I would get the chicken pox. Chickenpox is easier when you're 2 than it is when you are 82, okay? It can be very dangerous. Nowadays, they use active artificial immunity, which is giving somebody the vaccination, which is where you're actively giving somebody a shot, which is artificial because you're not naturally getting that infection. Passive, the difference between this, and these are the ones that students struggle with the most. Natural is something like a mother breastfeeding or antibodies passing through the placenta to the baby, okay? Artificial, this is where people get the most confused. If you are exposed via a needle stick, okay, to hepatitis B, they will give you an injection of hepatitis B antibodies. This is different than the vaccine that you got, which was the active artificial. They gave you a little dose of hepatitis B to make antibodies to it, okay? In this case, they're like, oh my goodness, you don't have the vaccine. Let's give you antibodies to try and kill off anything that may have gotten into your body, okay? So that would be giving somebody antibodies to fight off an infection. Very different from the active artificial, okay? So make sure you know the difference between those two. All right, that concludes our section on the adaptive immunity. I hope that helps you out a lot, especially some of those videos. If you have further questions, certainly let me know.